Okay, now we're going to move on to case study 2, which is on Acinetobacter baumannii. Now, this is a gram-negative bacillus, gram-negative rod. It is typically multi-drug resistant, and basically there's a couple of agents, colistin and tigecycline, which are reserve agents. But this is one of the organisms that's probably gone most towards pan-resistance, where there's really nothing in the drugstore that we can use to treat infections. That's the way it's looking as we're moving forward. It's typically associated with wound infections and other soft tissue infections, ventilator associated pneumonia. It can get into the bloodstream and cause bloodstream infections. And one particular feature in yesteryear was that it was often seen in military personnel returning from Iraq and Afghanistan. So both in the UK and in the USA, this was a feature of this particular organism. And in fact, it got nicknamed Iraqibacter at one stage. And in those settings, what we used to see was that military patients would then transmit, the infection would be transmitted from military patients to civilian patients in healthcare premises. And I'm going to give you a particular example of that from our own experience in a moment. Problem with this organism, it's hard to identify in the clinical laboratory. There are other, what used to be called genomo species, which are, you can't really do any any biochemical tests to distinguish them. They're now called Acinetobacter pittii and nosocomialis. And so it's tricky to tie this one down. And what was known at the time that we did our study was that there were what we call clonal outbreaks, so outbreaks caused by a single clone of bacteria spreading through one hospital or a series of hospitals, a series of wards. And these have been identified by various techniques where you looked at parts of the genome, particularly the most changeable parts of the genome. These techniques, pulse field gel electrophoresis, variable number tandem repeats, and various gene-specific assays. But at the time that we started this study, there was not so much known about the mode of spread and transmission chains going through the hospitals, and that hindered optimal management of outbreaks and rational design of policies. So, are we seeing particular spread in a very particular setting? Is this all over the hospital everywhere? If it's everywhere in the environment, your infection control policies are going to be different than if it's only in one ward and you want to concentrate on keeping it only on that one ward. Mechanisms of resistance may be hard to identify in individual cases, and we don't really understand much about pathogen biology, although I'm not going to say much about that in this presentation. Now, what I'm going to talk about is an outbreak that happened in 2011 and 2012 with what was known as the pulsar type 27. So when it was typed by pulse field gel electrophoresis, this was the type 27 that was seen. And this was first reported in the hospital in Birmingham, where I used to work, in July of 2011, in a military patient, I think, returning from Afghanistan. And it had been typed by what was then called the HPA, now we call it the UKHSA, and designated this unique pulse type 27. The outbreak in that hospital, Queen Elizabeth Hospital in Birmingham, lasted for 80 weeks and involved 60 patients, including both military and civilian patients. So it was a substantial and troublesome outbreak. What we did was we got 91 isolates from patients and the environment, and we genome sequenced those on the Illumina MiSeq, a desktop sequencing instrument. And in many cases, we sequenced multiple isolates from the same patient at different times or from different body sites. And the first thing we showed when we did genome sequencing was that this outbreak strain was different from all the other well-characterized strains which had their genome sequenced at that time. And that included strains that have been implicated in previous outbreaks in our local hospitals. So we knew this was a new thing, but it hadn't been seen before by us or by the public health authorities. And this has been written up. If you're interested to know more, you can look at this paper that came out 10 years ago. Jackie Chan was my PhD student at the time. Her and Mike Halachev did the work on this, most of the work on this, but there was a team of people also working on this from the hospital, included my colleague collaborator Beryl Oppenheim. And so we basically did whole genome sequencing on all these isolates, and we found a number of genotypes. And the first thing that was quite rewarding was actually kind of made sense. So when we mapped the various single nucleotide variants or SNPs onto the passage of time, we saw that the initial genotype, the reference genotype, where there were no SNPs, because that's what we used as a reference, was there at the beginning. As we accumulated more SNPs as time went on, we kind of went down a pathway. So genotype 2 had a couple of SNPs that separate from genotype 1. Genotype 4, there was a larger number of SNPs there, 13, I think it was 14. Genotype 3 was off there, that had 6. And as time went on, there were more SNPs. So as we get towards the end, there were many more SNPs compared to the first genotype than there were the intermediate genotypes. And you kind of get this hub and spokes approach where there are major genotypes, where there are lots and lots of genomes all identical, and then you get just variants where one SNP difference or a handful of SNPs different from those as we go along. And I should point out that the numbers there are not the numbers of SNPs. They're actually, I misinterpreted that myself. They're actually the SNP numbers. So SNP number one, SNP number 2, so in fact, I misspoke. Between genotype 1 and genotype 2, there's one SNP. Between genotype 2 and genotype 4, there are actually two SNPs. There's SNP 5 and SNP 9. Now, when we map this onto time and space, we saw interesting and interpretable patterns here. So we looked at the wards. They're coloured by the wards in the hospital, and they're coloured by the weeks in the outbreak, and these are the individual patients. And we can see that we started off in ward 1 with this genotype 1, and we can see that, two patients there at the beginning had identical strains at the same time. So it's easy to posit that what was going on was spread within that ward from one patient to another. And in fact, that happened for some time. You can see that ward 1 actually was the predominant ward for most of the outbreak, and the genotypes spread within that ward. So in genotype 2, you can see a series of serial cases of patients with that particular genotype. And what you can see, though, is that some of these patients move around. So that patient 11 moved from ward 1 to ward 3. And then a little while later, we start to see on that ward 3 more isolates that have no previous history with other genotypes. So genotype 3 has moved across there. As we get towards the end, a particularly striking feature emerges, though, which is that we saw lots of patients on the burns unit, the ones shown in red here. And this was a bit puzzling, so let's focus in on that in more detail. So what we see here is we see these multiple patients, all coloured in red. Now, if you're looking at a normal ward, you've got multiple patients in beds on that ward. They're typically not in all the patients on one big unit. They're typically like 4-bedded units within a ward. But it's very easy to imagine that patients move around in the ward, pop out to the toilet or something. They contaminate the environment around them. A nurse may go from one patient to another very quickly. It's easy to understand that, you can get transmission within that environment. But on the burns unit, each of the patients was within their own separate room with their own ensuite toilet facilities. And so we were really quite puzzled as to how is it that this particular multi-drug resistant strain is being transmitted from one patient to another within that setting. It took a while for the penny to drop, and what we realised was that although they were in separate side rooms within the burns unit, they were all going to the burns theatre to have their wounds debrided. So what happens in a burns patient is that typically all the dead and necrotic tissue in the burns is removed to prevent scarring and also to prevent infection. And so the common link was all these patients were going to that burns unit. And what happened was we then sequenced isolates from the burns unit from there, and we found that there were actually isolates from the burns unit itself that actually had the same genotype. And so it was in the burns unit that this was where it was being transmitted. And once that was realised, you can see there's that red arrow. There was a deep clean went on, and they actually eradicated the infection from the burns unit. And there were a few patients still in Ward 1, still going on there, but that was brought under control as well. And that marked the end of the outbreak, both in the burns unit and luckily in Ward 1 as well. So this is an example where having the genome sequences and knowing that the genome sequence was telling you that transmission of a particular strain, a particular clone was going on before your eyes in a particular environment meant you focused down your attention to that environment. In fact, there was also transmission that happened between two patients on a ward, and it was like, what's going on here? And it turned out they'd used the same bed. They used a particular burns mattress, and that had also accounted for the fact that got contaminated, and that's how it was transmitted. So having this very fine-grained view of the genomes allows you to really dial down into your understanding of what's going on and your ability to control infection. So that is all I'm going to say about that particular outbreak, where genome sequencing was actually informative. I haven't said anything about the actual drug resistances that was seen in that strain, but if you want to do your own student participation here, again, as before, go to that assembly website of NCBI, search for Acinetobacter baumannii pulsar type 27. That should take you to an assembly, a reference assembly from our outbreak. As before, download and unpack that genome assembly and upload the file to ResFinder. Here there isn't an option to select Acinetobacter baumannii as a species, just use the default and then see what you get. So if you now spend a few minutes doing that, we can pause this now and end this particular video, and you can start the next one when you've done that particular exercise.